All right, welcome back everyone to the scientific sessions of the PMSF We Are Family Conference. We have a great session today focused on gene therapy. And as you can see, we have a bunch of panelists today, so we're really excited to have a jam-packed session. And I will introduce speakers right before they speak, and we will start with an introduction from myself and Geraldine Bliss, and then we will have a short talk by Dr. Gouping Feng. And then we have a couple of representatives that you see here from Jaguar Gene Therapy who will go next, and then we'll return to Dr. Feng for a final part, and then we will take questions for about 15 to 20 minutes. So without further ado, let's get started. I will share a brief word from our sponsor, who is Jaguar. So just 1 minute as I pull that up.
Our mission at Jaguar is to accelerate breakthroughs in gene therapy for patients with severe genetic diseases.
With gene therapy, we can give the opportunity at a better life. That's what makes Jaguar special.
That we've been really fortunate at PMSF to be actively involved in this gene therapy approach long before a clinical trial would ever start. We've been meeting with Jaguar every month for over a year, and we've been really thankful to be given a seat at the table with them. We're not always able to share this progress before a program is announced publicly, but we wanted you all to know that these conversations are happening. We don't feel it's our role to directly promote any drug development pipeline, but we do feel it's our role to stay informed and to offer feedback to companies on things we think you'd want to know, things that bring you hope, challenges you may have, and things that might concern you. Another big part of our role is to share data that helps companies be invested in our syndrome. So a big thank you to you all who participate in the International Registry/Data Hub and in the Natural History Study. While it's partly our mission to support you always with your immediate medical concerns so you can get through the day-to-day, it's also our goal to strategize for a better future in drug development, and we're interested in continuing to do this by funding clinical research, being aware of current technologies like gene therapy, networking, and helping you tell your story to find earlier markers and better assessments. And we've been working alongside CureShank on a couple of these things, communication with Jaguar being one of them. So rather than having separate yet similar seminars explaining these things, we're happy to be combining this into one. And I'd like to give a moment for Geraldine to also say a few words.
Great. Thank you so much for inviting us to take part in this. My connection to Jaguar gene therapy began with an introduction from Dr. Feng. He was one of the first scientists to develop a mouse model that was missing the SHANK3 gene. Over the next few years, he discovered that the re-expression of the SHANK3 gene in adult mice led to improvements in behavioral abnormalities and neural function. And this breakthrough discovery suggested that PMS could be reversed through gene therapy. So in 2019, Dr. Feng told me that he was making very good progress in developing a gene therapy approach for SHANK3, and he shared the exciting news that there was a venture fund that was interested in advancing the research toward clinical trials. A few months later, Dr. Feng introduced me to the team that would eventually become Jaguar Gene Therapy. And since that time, we've had regular updates with the Jaguar team. At every step in the process, they've stopped to ask us questions about the lived experiences of individuals with PMS. Their team has expressed a genuine interest in our children, and I have absolute confidence in Jaguar's patient-centered approach. It has been a delight to work with the Jaguar team, and they have truly been a model industry partner.
Thank you so much for that, Geraldine. I think we're both thrilled to be, working with Jaguar and having and Gouping and having a seat at the table with them. That's a perfect segue to introducing Dr. Feng. He is a professor of neuroscience at MIT and directs research at the Broad Institute of MIT and Harvard. His lab is devoted to understanding synapses, which are the spaces between neurons where they communicate. He studies the development of these synapses normally and their dysfunction during disease. Dr. Feng has done foundational work studying SHANK3, which, as we all know, is an important synapse gene often affected in PMS. He has generated animal models, including mice and non-human primates, which have altered SHANK3 and allow models of PMS to be widely studied in laboratories. He's also involved in developing technology and overseeing projects surrounding SHANK3 gene therapy, and we are so thankful he could be here today. Welcome, Dr. Feng.
Thank you, both Kate and Geraldine. And first of all, I want to thank you for organizing this meeting, but I also want to thank the families for their support. This is critically important for encourage our scientists, especially young scientists, get into the field to try to develop understanding of the disorder, but also develop therapeutics that eventually we hope will help families. This is very inspiring for young people getting into the field. When they see their research may one day benefit people. So this is a great time and great opportunity. So I was asked to give a just brief overview at the beginning. So I want to make it quick and simple. Okay.
Yeah, perfect.
All right. So I'm focusing on, because there are many different ways to develop gene therapy for different disorders. So I'm going to mostly focusing on monogenic neurodevelopment disorders. So because of the genetic advances in genomic sequencing, and we now getting more and more clear pictures. So basically, there are about 25% of individuals with ASDs, especially severe ASDs, have identifiable causative and protein disrupting rare genetic mutations. This have been, proved again and again in many large-scale genetic studies. So for these patients, which SHANK3 mutation or Phelan-McDermid syndrome is one of them. And for these patients, there are possibilities that we can develop gene therapy. But we also have to understand most of ASDs are polygenic, and we don't have a clear single genetic cause. Usually it's a combination of many factors. So for these, we still need further studies and develop different approaches or different strategies to develop therapeutics. And for most of these monogenic disorders, usually most patients have very severe neurodevelopment defects and intellectual disability. ASD and autism, phenotype is only one of them. There are many other, symptoms or abnormality for these patients. One of the things people always asking, and we have been also, studied in the field, the neuroscience field, is if you have a single gene mutation, why cannot we just go develop a drug that treats this disorder? Because you know the cause of it, right? You know the gene, and many cases we know what this gene does. So the issue is for most of the monogenic disorders, each gene actually play many different functions. They involve many different pathways. The SHANK3 is a perfect example. SHANK3 interact with more than a dozen other proteins to perform its function. So we call the SHANK3 a scaffolding protein. Basically, it forms a scaffold, brings things to the neuron communication site we call the synapse. So it's almost impossible. It's really difficult. You can develop a strategy or find a small molecule that can treat all different pathways because they have different functions, right? So that's why we call it a monogenic disorder, but pathologically, they are actually not monogenic. There are many pathways. So then how do you, develop effective treatment for this kind of disorders? So after decades of study, we gradually realized, and with the technology development, maybe the best way is to directly go to the root cause of disease, which is the genetic mutation. So for monogenic disorder, which means single gene mutations, now we have many different ways to develop gene therapies to correct the genetic mutation. If you correct genetic mutation, then many of the downstream pathways, if not all, will be corrected automatically, right? Because the dysfunctional gene has been, the function has been restored. So that's why gene therapy provide a potential very effective treatment or even a cure for these monogenic disorders. So there are some general consideration of gene therapy approaches. There are many new technologies that have been developed. For example, you probably all heard, a new genome editing technology, including CRISPR, right? Including base editing, RNA editing. These are holding, great promise for gene therapy, but there are still very significant issues such as off-targeting, immune response, efficiencies, and many other things to be resolved before they can be widely used clinically. And I'm not getting into detail. This is the future of gene therapy. But there are other technologies such as gene replacement, gene expression regulation. They are much more mature, right? So after decades of research and development, now it's primed for clinical use. So these are in the next at least 10 years, you will see a lot of them in clinical trials or even develop into therapy for clinical applications. So since many of the Shank3 patients have large deletions, including Phelan-McDermid syndrome or large small deletions, and they are heterozygous, that means patients still have one copy of normal gene, right? So in this case, gene replacement and gene expression regulation are particularly suitable for Shank3 patients and for many neurodevelopmental disorders. So the other thing is, unlike many other tissues like liver, kidney, lung, heart, muscle, delivered gene therapy vehicles into the brain is very difficult because the brain is protected from other, hazardous, factors. So there is called a blood-brain barrier. Whatever in your blood, good things needed, energies, small molecules can get into the brain. But we also prevent that all these toxic things are attacking to the brain. There's a barrier. This barrier is very tight. So how do you get the gene therapy thing you develop and get into brain? So right now, there are many new technologies are being developed, but they're not primed for clinical use yet. Right now, the most effective way to deliver genes or gene replacement or gene regulators into the brain or even genome editing technology into the brain is still adeno-associated virus. And AAVs are widely developed in clinics. There are hundreds of clinical trials being used. It's very safe. And it's not saying it does not have any problem, but it's quite safe so far in clinical use. But that doesn't mean we are ready tomorrow to go for use gene therapy for brain disorders right now. There are still main issues. In my view, three main technical issues we need to be resolved and to be solved before each gene therapy, and not all three will be applied to all the gene therapy. But for Shank3, for example, AAV has a limited packaging size, right? And Shank3 is very big, and some of the transcription gene regulation is also very big. So how do you get this packaging into AAV is currently an issue. The second issue is many genes in the monogenic disorders, most of them are loss of function, right? Loss of one copy, loss of the function of the one copy. And so you want to replace the function. But overexpression is also a problem. We could potentially cause toxic effect, safety issues. Also, most of genes don't express in all the cells, all the neurons in the brain. They have very specific expression patterns, specific cell types. So we needed to express the same gene in the same type of cells that originally endogenously expressed. So ectopic expression can also cause safety issues. And the third thing that I talked about, how do you effectively deliver the AAV and carry your gene editing or gene replacement things into the brain? So these three things we need to solve, right? So that's in general for almost all the gene therapies. But for Shank3 mutation, most of you know really well, right? And it was originally identified as a major gene deleted in Phelan-McDermid syndrome, which is deletion of chromosome 22q13.3. And patients have developmental and speech delays, severe intellectual disability. Some have seizures, sleep abnormality, low muscle tones, and also autism spectrum disorder. And so currently, it's quite clear the loss of Shank3 gene in the 22q13.3 region is the main cause of neurodevelopment defect. So the idea is if you replace or restore the function of Shank3, even in Phelan-McDermid syndrome, which sometimes have more than Shank3 deleted, you probably will benefit significantly in regarding to neurodevelopment disorder, neurodevelopment defect. And also, there are many studies, clinical studies have identified nonsense and missense mutations. There are many of them are loss of function, right? So there are many different mutations in ASD patient with SHANK3 mutation. There are symptoms and are similar, quite similar to 22q11 3.3 deletion in Phelan-McDermid syndrome. Further strengthen the argument that SHANK3 mutation and SHANK3 deletion is the main cause of neurodevelopment defect in Phelan-McDermid syndrome. So what is SHANK3? SHANK3 is a family of proteins. There are 3 SHANKs, SHANK 1, 2, and 3. And they are structurally and functionally very similar. They just expression patterns and are quite different. So SHANK3 actually is a key postsynaptic protein. So basically, it's critical for neuron-neuron communication. So they are at the excitatory synapse. There are 2 types of major synapses in the brain. One is the excitatory synapse, one is inhibitory synapse. They are very balanced to work against each other. So we have balanced the neuronal activity. Now, loss of SHANK3 leads to loss of many signaling proteins at the synapse. Basically between neuron-neuron communications. And so because the scaffolding protein, so it does not, it's not like enzyme, a kinase or channels. It does not function at the synapse alone. The function is by bringing many other proteins, anchor this protein at the synapse so neurons can communicate properly. So it's not surprising if you lose a SHANK3, you will have many neuronal functional problems because neurons cannot communicate properly. So the current estimation is SHANK3 mutation account about 0.5 to 1% of all ASD patients, and in severe ASD patients with intellectual disability may account for about 2%. This number can change with in the future with more and more screenings and larger scale studies. So what do we need to consider if we are trying to develop SHANK3 gene therapy? So there are some specific considerations, right? So, and as you probably know, many of the SHANK3 patients have large deletions and they're heterozygous. Therefore, with large deletions right now, we don't have a genome editing new technologies easily replace them. So gene replacement and transcription regulation probably are particularly suitable because they're heterozygous, they have a one copy of one normal copy. And SHANK3 also is quite big, the genes are quite big. And with the promoter enhancers is significantly exceeded the AAV packaging size, right? So we need to develop a smaller functional, we call it mini gene, I will talk about later. And also, SHANK3 duplication have been identified in a few patients with ADHD, Asperger, or bipolar disorders. And transgenic mice, overexpressing SHANK3 in mice, already show there is a hyperactivity, abnormal synaptic activity, or even seizure. Therefore, control SHANK3 gene expression is very important. You cannot just put a SHANK3 gene in, overexpress it. So how do we control it? That's the second issue we need to solve. And the third thing we need to consider is SHANK3 is widely expressed in the brain, but not in all the cells. We don't know whether you express SHANK in ectopic places or cells where that cause a problem. So we want to not only know that, ideally develop a system that only express SHANK3 in the cells that endogenously express SHANK3. So like for many neurodevelopment disorders, SHANK3 actually is important for neurodevelopment. So the question is, what age is the most effective if you have a effective gene therapy? So we need to determine best developmental stages for gene therapy. We use transgenic mice. We showed that in adult it can rescue some certain defect, but in early on, postnatally, early on, it can rescue almost all the abnormalities. So we need to determine that what is the most effective, therapeutic window for patient. And also right now, there's no way, at least no technology right now, we can deliver even AAV into all the cells we want to deliver. So what's the percentage of cell do we need to correct that it will have a therapeutic effect? right? So all these things we need to develop. But the good news is they are really now progressing in all these aspects, especially for SHANK3 and some others. So we have now, I'm going to just, this is I already talked about. I'm going to just summarize. I will give you some details later on. We now have developed a mini gene which can be packaged into AAV with very high titer. And we also demonstrated that this mini SHANK3 gene can rescue neuronal defect when we put it into the mouse brain. We also showed that this mini gene, if we put it in the mouse brain, can rescue all the behavior abnormality we can measure in a mouse. So there is hope that if we continue to develop this and testing the safety, maybe in the near future we could put it into clinical trials and with the potential to benefit the patient with the SHANK3 mutations. So I'm going to stop here.
Thank you very much, Dr. Feng. That was fantastic and very informative. I have a feeling you already addressed a few questions that I know we've received previously, so thank you very much for that. So next, I will introduce both speakers right before they speak, and then they can switch off presenting. So we have a couple of representatives from Jaguar Gene Therapy. First is May Tobar. May is the Director of Patient Advocacy at Jaguar Gene Therapy. She has extensive experience in global strategy development, marketing, and product launches. She previously served as Director of Global Marketing at Advocacy, Novartis, supporting the launch of the drug Zolgensma for the treatment of spinal muscular atrophy, one of 2 recently approved gene therapies in the US. She's passionate about developing connections with patient communities. And second, we have Dr. Dan Gallo. Dr. Gallo is the Vice President of Medical Affairs at Jaguar Gene Therapy. Dr. Gallo has led numerous US and global medical and cross-functional teams in support of drug launches and rare disease space. Before joining Jaguar, Dr. Gallo served as Vice President and Head of the US Medical Affairs at Orphazyme AS and has over 10 years of experience in drug development and medical affairs. Welcome to both of you.
Thank you so much, Kate. You can switch over to the next slide, Dan. I will start out by just telling you what we're going to talk about today. So I will give you a super brief overview of who Jaguar Gene Therapy is and what we're working on. And then I'm going to kick it over to Dan. Dan is going to talk a little bit about our approach to gene therapy. So Dr. Feng mentioned a couple of different types of gene therapy that are out there. Our focus for our JAG201 program is on gene replacement therapy. So we'll focus on that. We'll also talk a bit about clinical development for gene therapy specifically. There are some unique differences between gene therapy, drug development, and also traditional drug development. So we'll walk through that. And then, of course, we will talk through our investigational gene therapy. It's JAG201 for disorders of SHANK3 mutation or deletion. So who are we? We are a relatively new company. We started like under the radar, I guess is a good way to say it. In 2019, we did announce ourselves to the public in February of 2021. And very soon after that announcement, we reached out to both Geraldine and the Phelan-McDermid Syndrome Foundation. So we're very proud to be partnering with both of these organizations and have been essentially since we have been announced as a company. So a little bit about gene therapy on the next slide, Dan. Our mission is very simple. We want to accelerate breakthroughs in gene therapy for patients living with severe genetic diseases. So that word accelerate is used very deliberately at Jaguar. We want to make sure that we're accelerating treatment options for communities like the Phelan-McDermid syndrome community where there are not enough options for you. And we also think that we are in a unique spot to be able to do so. We have a lot of our leadership team, a lot of our team members, myself included, as Kate mentioned, we're members of the Advocacy team, where we were part of the team that did the development and the launch of an AAV9-based gene therapy for children with spinal muscular atrophy. Developing a gene therapy is complicated. Dr. Feng mentioned a lot of the challenges and complications that can happen with gene therapy drug development. And we think that we're in a unique spot in the fact that we have in fact done it before. Another thing on this slide I want to mention, we say that there's a patient and a family at the center of everything that we do, and I want to just talk about what that specifically means for us. What it specifically means for us is our partnerships with organizations like the PMSF, with CureShank, and frankly, with families like you. We do not know and will never know as much about Phelan-McDermid syndrome as you do. You're the experts, you live it every single day. And we feel very strongly that in order for us to develop and accelerate gene therapy options, we must listen and learn from you to be able to do so. So moving on to our next slide, what we actually are doing. We have 3 gene therapy programs in development right now at Jaguar. We have JAG 101, which is for type 1 galactosemia. Skipping to the bottom, we have JAG 301 for type 1 diabetes, and certainly what we'll spend the most time focusing on today is JAG 201. JAG 201 is focused on the SHANK3 gene that Dr. Feng talked about. So we're going to be targeting SHANK3 mutation-related conditions that of course includes Phelan-McDermid syndrome, ASD, and other severe neurodevelopmental disorders. I think one thing that's very important to mention about this slide is that all of those little green and blue bars stop at that preclinical phase. Basically, what that means is that we are not currently in the clinical phase of our drug development, which means we are not having any trials in humans. All of our trials right now are in the preclinical stage, so we're testing in animals, and during this stage, we're learning a lot about each of these, including JAG 201, and certainly Dan will be able to share a little bit more about that. So with that, Dan, I'll kick it over to you to talk about gene replacement therapy.
Thank you, May, and thank you, Kate, for the introduction. Can you all hear me okay? Excellent. So thanks for this opportunity. I'm really happy to be able to connect with you all and talk directly to you all, the patients and the families and the advocacy community. We have a couple of topics that we're going to cover here, and we're going to start off with our approach to gene replacement therapy. So as Dr. Feng mentioned, there's numerous ways to deliver genetic function. There's numerous ways to approach gene therapy. At Jaguar, we're focused on gene replacement therapy as a potential treatment for genetic diseases. With gene replacement therapy, what we're aiming to do is to deliver a functional copy of a gene that's otherwise defective or missing in an individual, but deliver it back to the body, to the target tissues, so that we can ultimately restore the production of the protein, as well as the natural function of that protein to the cells that are affected. In this way, we're aiming to address the root cause of the disease by replacing that missing or defective protein and hopefully change the course of the disease. As May mentioned, this is a therapeutic approach that's being investigated across multiple diseases. There are many gene therapies that are already in early preclinical research, as well as clinical research and clinical trials. And as May mentioned, there are some that have already been approved, including one that many of the folks on our team have worked on. So when we talk about gene replacement therapy, there's, again, a number of ways to deliver a new or introduce a copy of a functional gene. The approach that we take is the one that Dr. Feng mentioned, which is using an AAV vector or an adeno-associated viral vector. And so I want to start off just by highlighting what a viral vector is, how it's used to deliver genes, and how a viral vector differs from a virus. So on the left side, we have a cartoon diagram of a virus, and this highlights sort of the key components of the viral structure. Viruses are biological entities. They're not alive on their own, but they multiply inside of a host, which can be a living organism, a human, animal, or plant. They have a number of key components. So at the center of the virus, what you see here is essentially a cross-section through the center of a spherical virus. And at the center of them, what you'll see is a viral genome. This can be either DNA or RNA. And then importantly, just outside of that, you'll have a protective coat. This is what we refer to as a capsid. This is made up of protein. So again, we're looking at a cross-section through it, but you can almost imagine this is a three-dimensional structure, a protein shell surrounding that DNA or RNA, and it serves as a protective coat, protecting it from the environment. Some viruses also have an envelope, which is just a layer further outside from that protein, and they may have additional proteins embedded with that envelope that in some cases can be used for entry into target cells. As I mentioned, viruses, they're not alive on their own. They survive by replicating inside of host cells and hijacking the cellular machinery within the host cell to read that genetic code, to make new proteins, make new components of viruses so that new viruses could be assembled. Now, with viral vectors, what we're doing is we're basically leveraging some of those features of a virus to use a virus or a viral vector as a delivery vehicle. So when we talk about viral vectors, what we're talking about are taking what would otherwise be naturally occurring viruses and removing any disease-causing or replication-allowing genes, and we're rendering them non-pathogenic if they're not already non-pathogenic. So adeno-associated virus is an example of a virus that's generally thought to be non-pathogenic, but we're further making it non-pathogenic by removing the ability for it to replicate and removing the ability for it to make its natural viral proteins, and we're making it replication defective. In gene therapy, then what we're doing is taking the functional gene of interest. In our case, for our discussion today, that would be a functional version of the SHANK3 gene, and replacing it within the genetic structure, the genetic content of that virus, so that it can be incorporated and housed within the capsid or that protective shell of that viral vector. In this way, because the viral vector already has its own mechanisms for being able to enter the cell and deliver genetic material, we're basically leveraging that function to deliver our gene of interest to the nucleus. Now, depending on the type of vector that's used, the functional gene can exist separate from the DNA, so outside of the host DNA, but within the nucleus. This is generally what happens when we talk about AAV vectors. Or alternatively, you may have heard of lentiviral vectors. This is a different type of vector, but one of the key differences here is that the genetic information from these viral vectors carry are ultimately inserted into the host DNA. So why have we decided to use AAV vectors? So again, the AAV adeno-associated virus vector platform is one that's well characterized, and in some instances has been proven. As we mentioned, there are approved gene therapies that are based off of AAV. AAV is a vehicle that can be used to deliver functional genes into the cells, right? The target cells, which is what we're aiming to do here with SHANK3. They are an effective delivery system because, as we mentioned, they are non-pathogenic, but they have very efficient ways of gaining access to target cells. We think that it's beneficial that they primarily exist as episomes and they don't integrate into the host DNA. And again, the goal here is to deliver a working version of the faulty gene, ultimately helping to restore the production of natural protein or enzyme, so that ultimately the cells that take up that DNA can be restored to function. And with AAV, there's the potential that with a single treatment, a one-time administration, you may be able to provide a lifetime of benefit. But in addition to leveraging this platform, at Jaguar Gene Therapy, we are very focused on ensuring that the preclinical studies that we design are robust and allow us to fully evaluate the benefit risk profile of the therapeutic, the investigational treatment that we're developing, so that at the end of those studies, we can analyze those data and make a thorough evaluation of the potential benefit risk profile of that AAV for evaluation for entry into human clinical trials. In addition, while we are currently focused in our three pipeline programs, while we're currently focused in AAV, we are always evaluating next generation technologies, including some of the things that Dr. Feng mentioned, for future programs, including new vectors, new vehicles, and new delivery methods. So I'm going to shift gears here and talk just a little bit about clinical development for gene therapy. And when we think of drug development, there are many phases, as we can see from the schematic, and you may be familiar with these, right? But early in development, you have your discovery phase, you have your preclinical phase. And what this is highlighting is that as you move through development, fewer and fewer drug candidates end up making it or progressing through to later stages. In the preclinical stages, as we'll discuss, we're really looking at evaluating how an investigational treatment behaves in animal models. So we'll ask questions about safety, efficacy, tolerability, toxicity. Once you have enough of a profile that supports, from a benefit risk perspective, moving into clinical studies, you would then submit an IND seeking permission to initiate clinical trials. Once you enter clinical trials, there are multiple phases. So in phase 1, these are generally smaller studies, oftentimes done in healthy volunteers to evaluate safety of investigational treatments. By phase 2, these patient populations evaluated in the studies increase. You ask additional questions about safety, potentially additional questions about dosing, and start to look at efficacy. And by phase 3, ideally, you've selected the appropriate doses for investigation in humans, and you're looking primarily at the efficacy and safety profile that you will use to build the application for review by the FDA. Now, depending on the type of investigational treatment you're developing, that can be a new drug application, or in the case of gene therapies, that could be a biologics license application. So the idea here is that's what's submitted to the FDA so that that efficacy and safety profile can be evaluated. And within half a year to 2 years, the FDA will review that and make a decision on whether or not it reaches approval for marketing. And so a couple of things I want to highlight with JAG201, as May mentioned, we are currently in the preclinical stages. So right now we are conducting the research, gathering the data that will allow us to make that benefit risk assessment of JAG201 based on how it behaves in animal models. And so that's where we are today. One additional difference from what I just mentioned is that when we think about clinical trials for gene therapy, oftentimes they may take a little bit different trajectory. And there could be a number of reasons for this, right? But one in particular is that, a lot of times in gene therapies are evaluated in rare diseases. And this generally means that there are fewer individuals affected by these diseases. And so oftentimes what this means for clinical trials is that the clinical trials are smaller in terms of the number of patients that they enroll. And oftentimes you'll also see that the phases of the trials can be combined. So in some cases, your phase 1 or your phase 2 may be combined, and those may actually serve as registrational trials, which support the submission of your biologics license application. So the timelines can vary a little bit in gene therapy. So with JAG201, we are currently in the preclinical phase, and I've already spoken to some of this, but again, in the preclinical phase, what we're looking to do is really to build that benefit risk profile and understand what that benefit risk profile is of that investigational treatment in animals so that we can make an informed decision on whether or not we should proceed in the clinical trials. Usually this involves evaluating an investigational treatment in multiple species. These could be rodent species or non-human primates generally, but potentially others. And these studies provide preliminary evidence of efficacy. So how well is the treatment working in a representative animal model? And based on the outcomes that you're seeing on those animals, can you start to make some assumptions about whether or not that might be translational into the clinic, right? Does it give us an idea of what we could expect to see in humans? We'll look at toxicity. So are there any harmful side effects that we're seeing in the animals that are being dosed with the treatment? Based on the different doses that we're applying, can we define a maximum dose that is tolerated by the animals? Can we define a minimum dose that is shown to be effective in animals? In every study that we do, we'll be capturing safety information to understand what is the safety profile when we introduce this gene therapy. And with gene therapy, we're also looking at biodistribution. So we ultimately have to administer the gene therapy. There are numerous ways to administer a gene therapy, but I think the important question is, can it get to the target sites? In our case, we're talking about SHANK3. And as Dr. Feng mentioned, we want to be able to express that SHANK3 in neurons. So are we demonstrating that we can appropriately deliver it to neurons and to enough neurons that we would anticipate that it would have some, a potentially translational clinical effect? So we do look at a number of dose ranges in preclinical studies. And again, the outcome of this is hopefully to inform the best doses to explore in human clinical trials. So I'm going to shift gears here and just talk a little bit about where we are with our JAG201 treatment, which is an investigational gene therapy for the treatment of SHANK3 mutation or deletion. Some of this was already spoken to with the introductions from Geraldine and from Kate and from Dr. Feng. But just to maybe re-highlight this. So JAG201 is an investigational gene therapy, again, for certain genetic causes of autism spectrum disorder, Phelan-McDermid syndrome, and or other neurodevelopmental disorders where there's a SHANK3 mutation or deletion. With JAG201, what we're aiming to do is to deliver SHANK3, a functional version of the SHANK3 gene, leveraging the AAV9 vector to address the underlying SHANK3 haploinsufficiency. Again, we want to enable expression of the SHANK3 protein and hopefully restore synaptic function in these cells that are affected by SHANK3 haploinsufficiency. It is designed to be a one-time treatment, so a single administration. And the JAG201 program, as alluded to earlier, is exclusively licensed from the Broad Institute of MIT and Harvard. And so you're all familiar with this, and Dr. Feng spoke to this, right? But when we talk about SHANK3, we're talking about individuals that have either a mutation in SHANK3 or a deletion in SHANK3 that results in haploinsufficiency. That SHANK3 naturally functions as a scaffolding protein. It has many interacting protein partners within the neuron that allow for synapse development and synapse communication. And so when you have a defective copy and you have that haploinsufficiency, you have reduced synaptic function. Our goal here is to be able to deliver that JAG201 to be able to restore SHANK3 expression in the cells that are affected by haploinsufficiency. And again, ideally by addressing the root cause of the disease, be able to restore some of that synaptic function. And so where are we today? Well, as I mentioned, we are currently in preclinical studies evaluating JAG201 in animal models, so both rodents as well as non-human primates. These studies are ongoing, and the types of things that we're evaluating include JAG201 safety, whether there's target engagement. So do we have confidence? Can we demonstrate that SHANK3 is reaching the sites that we want it to reach and that it's having the function that we would want it to have within those neurons? Looking at biodistribution. So again, is it reaching the target tissues in sufficient numbers? Dr. Feng spoke to, one of the challenges being that we want to make sure that we're reaching enough neurons that this will translate into a benefit. And so this evaluation of biodistribution will help us assess that, as well as measures of behavior. So we want to make sure that not only are we delivering the gene therapy to the appropriate sites, but that it's having a biological impact and a biological benefit in the animals affected by SHANK3 haploinsufficiency. So as we come to the conclusion of these studies over the coming months, the next steps will be to evaluate those data, evaluate the outcomes of these preclinical studies, and really make an assessment of what the benefit risk profile is and whether the data that we're gathering, the data that emerge from these studies, again, in rodents, non-human primates, support advancing JAG201 into human clinical trials. So of course, our hope is that they do. And we're all very committed to this work and very anxious and eager to see the outcomes of it, and hopefully be able to share that with you at a future update. So with that, I will turn it back to May.
Thanks, Dan. So as Dan mentioned, hopefully we will have updates for you in the next, coming futures. And in order to share that with you, as long as you are following the Phelan-McDermid Syndrome Foundation channels or the CureSHANK channels or both, we are very proud to partner with them. Any news that is newsworthy for you, we will make sure to share with them, and they will make sure to share with you. You're also very welcome to check out our website, follow us on LinkedIn, and certainly we'll be here, after all of these presentations to answer any questions that you have today. But if you think of other questions and you want to reach out, please email patientadvocacy@jaguargenetherapy.com. That email goes directly to me, so you will probably be hearing from me if you email that, but we'd be happy to hear from you. Thank you.
Thank you both so much. That was really great. And I will now turn it over to Dr. Feng again. We heard a little bit about a mini gene and some of this preclinical animal data, so he will share a little bit of that.
Thank you, Kate. So I want to tell you a little bit about why we are so excited about this gene therapy program for SHANK3 and what else we are doing, try to move forward. So as I mentioned, and Dan also mentioned, SHANK3 is a big gene. We now made a mini gene and cutting to much smaller so we can package into. This is not randomly cutting, based on the structure functional studies done by many neuroscientists before, from my lab, from many other labs. So we're based on this knowledge, based on structure, based on many different design and testing, we develop a mini gene that can be packaged into AAV for gene therapy. So how do we know it's, we need to know whether this mini gene is functional or not. So what we can do is pack into AAV, this mini gene. Then with a tag, we can tell where this gene is expressed. So we can inject into the mouse through IV injection, and at a different ages to see which age you can rescue. I can give, then you can test their behavior, their function, and to see whether you rescue them. So we tried many different ages because we want to know what is the window in the life of mouse. So we did a postnatal day 0 and 2 between. That means they are just born a day or same day or a day after, 2 days after they're born. Then the postnatal day 7. 14 and 28. So we did a whole bunch of studies, and what we found is in most cases it can rescue everything, as we can measure in mice. And in P28, which is young, juvenile, and also we can rescue most of things. There are a few things we did not rescue. We think there are particular reasons, and so for example, we can use electrophysiological methods to measure the key function of this SHANK3 gene, which is synaptic transmission, which means the communication between two neurons. So the black is the control Y type, the red is the mutant mouse, and the green is the mutant mouse with gene therapy. You can completely rescue the neuronal function. And behavior, same thing. We did social interaction, we did anxiety-like behavior, locomotion defect, motor learning, and repetitive behavior. They all can be rescued. So that's very exciting to us. And what we needed to know is whether this expression of this mini gene causes problems. For example, seizure, right? People have shown in mice if we overexpress SHANK3, actually from before birth in germ cells, it actually can cause seizure. So we found that whether we inject in different ages or different type of mutant animals, we actually both P17, P7, P14, and P28, we did not see any seizure activity. How do we know we can measure the seizure? We use a different autism model, and we know it has seizure. You can see we can measure it. But we all the SHANK3, mini SHANK3 treatment, we did not detect any seizure. So in this sense, at least in mouse, this gene therapy approach of mini SHANK3 is safe. Of course, we all know mice are very different from humans, and so we need a next level test, right? So we need models bigger and more closer to human brain. So that means we need probably non-human primate models. So my lab have been invested many years of effort to try to develop SHANK3 model in the non-human primate. We now have this model, and one of the things we do at the MIT and Broad is use a marmoset. Marmoset is a very small monkey, which is originally from Brazil, South America, and we now have developed a mutant SHANK3 marmoset based on the human mutation with a deletion in one of the key exons, and these monkeys actually have phenotypes that we can detect easily. So in mice, we said in patients they're heterozygous. That means you have one normal copy, one deletion. But in mice, one normal copy, one deletion mice, there are not many phenotypes you can measure. They are almost normal. So that's the difference between human and mice. So we use homozygous. That means you don't have any SHANK3 protein, which is significantly different from human patient. But in monkeys, actually, we use exactly the same genetic mutation in humans, which is one deletion copy, one normal copy. So in heterozygous, we actually found that so we then develop a system that can monitor monkey behavior, marmoset. So this monkey are very small, and in their home cage, you can see they running around. We use computer machine learning, computer vision to actually track them. These dots are just tracking their different body parts. So you can monitor their behavior, then the computer will automatically analyze their data. So you can see they track very well. Now we can study them. Is there a difference between the normal Y-type monkey and the SHANK3 heterozygous mutant monkey? And I just want to show a couple of things. We have many, many different phenotypes. For example, you can put an iPad in front of a cage, right? And then they ask them to touch a blue square. They will get a marshmallow. They like a marshmallow. So they will all, this is a heat map. You can see all the monkeys are staying in front of. As soon as the blue blue spot showed up, they will touch it and they'll go get a marshmallow. And you can see both control and Shank3 mutant monkey like to do that. But if you suddenly switch, right, switch the imaging from blue blue square to a marmoset imaging, white type still will go touch it and get the reward, but the mutant doesn't like this picture. Actually, will retreat to the back. So this is a very clear phenotype. Can our gene therapy rescue this, right? And the same thing. You can have two cages next to each other; they cannot cross. But you put a tablet, and then the marmoset will focus on this, as I showed before. But then you introduce another marmoset, a new animal, into the next cage. Most wild type animal will be very curious, come over, leave the tablet, and come over to explore and interact social with this other marmoset. But for Shank3 marmoset, they will completely ignore the other animal. They were still focusing on this tablet, and so they are much less interested in social interaction with other animals. So these are just examples. Now we can actually, already did the gene therapy for these monkeys with two different ages. One is about 10 months old. 10 months old is also juvenile compared to humans, and the other is 3 or 4 months old marmoset. So is already in the way. They already be administered this AAV and Shank3 gene therapy. So we will have a complete data by the end of this year. So this will tell us, so far we have not seen any abnormal behaviors. So we want to see whether it's effective, whether there's any abnormal thing. So this will tell, our hope is this kind of study in monkeys will help guide us, help at least give some guidance to the design of future clinical trials. So I will stop here. I want to acknowledge a lot of people. This is a large teamwork with many professors, many laboratories, many institutions. Especially, I want to thank all the people support this work. This is quite important, not only for us, but hopefully for the patient. And I particularly want to acknowledge people who the donors who made this work possible, and especially Lisa Yan and Hawk Tan, donated most of the fund to establish the Autism Center research at MIT, and the Stanley Center by Stanley family for psychiatric research abroad, and many other institutions, agencies, including NIH, and individuals actually supported our work, made this very large effort possible. I'm going to stop here.
Thank you so much, Dr. Fang. I think it's very unique that we're able to get this amount of information at this stage in drug development and have an opportunity for questions. So we really appreciate that and all of the talks that have already happened. So we will transition to Q&A now. We already have a few great questions in the Q&A, and we will try to get through as many as possible. So the first one I will pose to Dr. Fang, and he can pass it along if he would like. And the question is, we've said that Shank3 is a very long gene with non-coding and coding regions. Could it be possible to miss something in making a gene with only known coding regions, knowing that other regions could be important?
Yeah, it's a very good question. So in this case, so most of non-coding regions are regulatory. Since our goal is to replace the protein, so we use we call the cDNAs, coding sequences that encoding the protein. And so we may miss regulatory regions, which is definitely true. But in this gene therapy case, we change the regulation. We control the regulation. So we put a new system to regulate the expression. So in this case, it's much less likely that the intronic region will be important. However, this raised another question, which is there are many different splice forms, right? So you basically splice these called non-coding regions out. So there are many splice forms. In our case, we only express a truncated, a small mini form of the longest cDNA. Now, there may be some unique function for other forms. Right now, we may not be able to rescue. But the major isoform seems from mouse study, we can rescue them all.
Thank you very much. The next question I will try to tackle myself, and anyone can jump in if they like, but it says, what's the difference between the Angelman syndrome gene therapy clinical trial and PMS gene therapy future clinical trial? I think the major answer to this is that the genes that are being targeted are different. So even though Angelman syndrome causes a lot of very similar features as Phelan-McDermid syndrome and many other related disorders, they often have different underlying genes, sometimes even on different chromosomes that may also function at the synapse or elsewhere, but can result in very similar things. So while the approaches are the same and the challenges are the same, the underlying genes are often different. Does anybody have anything to add to that? Okay.
I'll just add, if it's okay, in Angelman syndrome, I think the approach mostly has been what's called an antisense oligonucleotide approach, where what they're trying to do is wake up the paternal copy of the UBEA3 gene, right? It's there, but it's normally silenced. And what they're trying to do is unsilence that paternal copy. So it's a bit of a different disorder. And so the approach is a little bit different than you would use in Phelan-McDermid syndrome.
Thank you, Geraldine. The next question is, when you do open, this is a great question, we get this a lot. When you do open clinical trials, will individuals missing more than SHANK3 be considered?
Yeah, I'm happy to take that one. So I think two things, right? So I think when it comes to the specific inclusion criteria, enrollment criteria for a trial, a lot of that will be informed ultimately by the profile that we see from our animal studies. But I think the short answer to this question is that yes, we will be enrolling, or if we get to the point of evaluating this in the clinic, we will be looking at individuals with not only mutations but deletions as well, right? And so deletions encompassing SHANK3 may also, have deletions in other parts of chromosome 22.
Something that we sometimes don't always communicate, and this is essentially what Dr. Gallo is also saying, is that when we talk about loss of function of SHANK3 of the gene / protein, that is also encompassed in deletion. So if you have a deletion that includes SHANK3 and neighboring genes, and even large deletions with 50 genes lost, if SHANK3 is included in that, you would have what we call haploinsufficiency of SHANK3, which you probably see a lot of in our presentations. The next question is related to the data from Dr. Feng's slide, so I will pose this to him. The therapy did not create seizures in mice. However, were there any mice who did have seizures, or if you caused seizures before treatment and the seizure activity then stopped?
Yeah, that's another very good question. So we actually tried really hard, even try to induce seizure in these SHANK3 mice, but we couldn't see any seizure. So we can tell whether we can stop that. We know in patients, a certain number of patients has seizure. Now we are trying to see that in monkeys, whether hopefully the monkey will, and that's another difference between mouse models and humans. And we had some other collaborations with our group in China, the big monkey, the macaque monkeys. Actually, we see some seizure-like activities. The gene therapy also will start sometime soon. This is also supported by NIH to support the biomedical collaborations. And now we are doing EEGs in the marmoset. If we detect seizure in the gene therapy, we have a control group as a mutant without any gene therapy. So we will know soon whether we can detect the seizure or not, and if we do, whether the gene therapy rescued. It's a really good question. We really want to know, but we don't have an answer right now because of the difference between the mouse models and the human patients.
Thank you for that. That was great. The next question is, and I can also help answer this, but I will kick this to Dan also and May, but how can our children become involved in clinical trials once they begin to happen? Speaking on behalf of the foundation, we will absolutely let you know any developments in any, any major stages that are reached. And I'm sure the same is also true of CureShank. So similar to what's going on with the current clinical trial that we have with Neurin, we will let you all know about that and contact information for enrolling. But does Jaguar have anything else to add to that?
I would just say that if we are able to move into clinical trials, we will absolutely want to let you know. So it will definitely be something that we share with you and something that we're very excited to share with the community. So I wouldn't worry about not missing it, if that helps.
Thank you. And another question is, will slides be available for today's presentation? All of our sessions are recorded and available in our app for the conference, usually by around a week post the session. So that these will be available. And we have a lot of folks that watch after the fact who can't make these sessions. So even for our panelists, just know that there are many people that are also watching this at a separate time. And the next question is, when does Jaguar believe they will advance or could advance to clinical trials theoretically?
Yeah, so that's a great question. So a couple things, right? Very, very much JAG201 is a priority for us, right? So we're doing everything we can to, to move safely but quickly. But we are still early in development. We are still in preclinical development. And so our ability to move forward into clinical trials depends on the outcomes of our preclinical studies. We are, actively working on those. We hope to see readout from those studies, in the relatively near future. But there are many things that could happen, right? We could see those results and determine that additional studies need to be done. We can have discussions with the FDA and they can inform us that additional studies need to be done. So it makes it, a little bit difficult to speculate how soon. But I guess the short answer is we're working hard and, we'll try to get there as soon as and as safely as possible.
Thank you. The next question is a bit of a discussion. It says, it seems to me it is lower risk both of the failure and of side effects to do first clinical trials with people completely missing SHANK3 rather than people with pathogenic SHANK3 variants. The preclinical studies so far are only with 1 or 2 variants. This seems rather small compared to the 60+ variants among humans in terms of translatability to that group. Does anybody have any comments on that?
So I think at least with regards to JAG201, and maybe this maybe somewhat addresses the next question as well. We're really developing it for, again, patients affected by SHANK3 mutations and deletions, but that result in haploinsufficiency, right? And so there's numerous ways to get to haploinsufficiency. You can have a deletion where you don't express the SHANK3 protein at all. You can have, potentially a nonsense mutation that, while you may have most of the coding sequence there, you don't actually express any functional protein. So there's a number of ways of getting there, but ultimately, at least the way that we're thinking about it now is that it is being developed for patients affected by mutations or deletions that result in haploinsufficiency. Which is why, with regards to the question of the deletion, are we looking at different size deletions or different deletions that span beyond SHANK3? The answer is yes. Right.
Thank you very much. And that also somewhat addresses the next question, which says, what about translocations? This is a question that we get a lot, in that translocations do often result in a deletion or a loss of material, including SHANK3 gene. So this would also apply to those for the most part, I believe.
Yeah, and also, I'll just preface that that's at least how we're thinking about it now, right? I think ultimately the ultimate inclusion criteria will be informed by the outcome of our preclinical studies, and those final decisions will be made at some point in the future.
The next question is actually somewhat similar, asking if a deletion versus another type of alteration genetically could affect what treatment is essentially given. And I think this was answered with the previous question, that anything that causes a loss of function to SHANK3, also called haploinsufficiency, when you have one copy that's loss of function, is theoretically the target to be addressed here. And also asked, can you anticipate when therapies will be tested in humans, which we just talked about. And a new part of this is how will the age of the patient affect this, which is a really good point.
Yeah, that's a great question, and not one that we can answer at this time. Again, part of this will be informed by the preclinical data that emerges from our studies. And when we design those studies, we are looking at specific age ranges in rodents, with the goal of ultimately being able to make a case based on those outcomes for treating patients of a particular age. So those ultimately those decisions will be made at a later date based on the data that emerge from our studies.
Yeah, our hope is that our monkey studies will really provide some better estimation what is the most effective age.
And something that we talked about in our clinical trial session a couple weeks ago with Dr. Liz Berry-Kravis was that, theoretically targeting things earlier on in life is always helpful for trying to get to things, quicker in terms of brain development, but that potentially, and this is not unique to gene therapy, but in general, potentially older individuals could also benefit if the animal studies do give us some of that information, if it translates. Does Jaguar have an estimation as to when preclinical trial analysis should wrap up? I think this is similar to the clinical trial question, but it varies and it's hard to answer. But do you want to answer that again, Dr. Gallo?
So unfortunately, we don't have an estimate, but just to reemphasize that we are working as hard and as fast as we can. And when we do have an update to share, we will absolutely share it with you all.
Thank you. I don't see any other questions right now, but sometimes they start to come in a little bit at the end. So we'll give people maybe another minute to ask another question if they have any. But these were all great questions. There were a lot of them too. So maybe they were all covered. Will genetic reports determine who can be part of the clinical trial? If in the future with a clinical trial?
Yeah, excellent point. So yes, I mean, I anticipate that if the benefit risk profile from the preclinical study supports moving into clinical trials, that a component of the inclusion criteria will be, understanding the characterization of the genetics, right? So what specifically is causing the haploinsufficiency? Is it a deletion? Is it a deletion of a particular size? Is it a loss of function mutation? So the short answer is, if we get to that point, yes, I do think it'll be important.
Thank you. Any other last questions? Anybody who's nervous to ask a question, please do. Oh, this one's just a nice comment. Thank you all for the work that you do. You're a great source of hope and inspiration. Thank you to the panelists and both moderators. Great presentation. Let's all hope this train stays on the track. We all agree with that. All right, I think we are good. If anybody has any more questions, feel free to email me at kate@pmsf.org and we can ask the panelists here. And we want to thank them so much for taking their time. We really appreciate getting all this information, especially at this stage in the process. And it is, somewhat late in the evening. So thank you all so much. And we will see you next time. Thank you.
Thank you.
Everyone.
Bye.
Have a good night.